Please be seated.
I open this academic ceremony in which Musa Idris will defend the academic thesis, Neural Influence on Colorectal Carcinogenesis, Unraveling Neural Signatures and Tumor-Neural Crosstalk. I invite you to present a summary of your thesis and the conclusions of your thesis.
Thank you very much. Dear Prorector, members of the Corona, dear family, friends and colleagues, here and online. In the next 15 minutes, I would like to take you through the research that I have conducted over the past years. I will explain the most important findings from my dissertation, which is entitled Neural Influence on Colorectal Carcinogenesis, Unraveling Neural Signatures and Tumor-Neural Crosstalk. Okay, first I will start with a quick introduction about the field of science that I've studied, which is nerves and colon cancer. Colon cancer is still the second deadliest cancer worldwide. Almost 2 million people were diagnosed with colon cancer last year, while no fewer than 900,000 people have died because of it. Here in the Netherlands, last year, more than 8,500 people have been diagnosed with colon cancer, with more than 3,000 people have died as a result of the disease. Therefore, there is still much to learn about colon cancer and how to better treat it. So to combat colon cancer, advancements are required in several key areas. Of course, to prevent the disease from occurring is always better than treating it, but once the cancer starts, then we need early detection tests to identify the disease in its earliest and most treatable stages, and we need new therapeutic avenues to enhance treatment options and improve patient outcomes. All these challenges require better understanding of colon cancer and its colonic microenvironment. So I would first give a quick introduction how the colon is constructed and what is its microenvironment. So the colon looks like a tube where its lumen is exposed to the food passing through. The inner layer of this tube is the epithelium, which looks under the microscope as a wavy one line of cells. Beneath the epithelium, still in the mucosa, what you see now, we find other cells like immune cells that are ready to interact against any intruder into the body. Among these immune cells, we know like B cells, T cells, macrophages, etc. Under this colonic mucosa, we find two muscle layers, and between these muscle layers and the mucosa, we find the enteric nervous system. This enteric nervous system is a complex network, consists of two plexes, the submucosal plexus, the high one, and the myenteric plexus. And this network consists of neurons and glia. This ENS coordinates almost all functions of the gut, like intestinal movements, nutrients absorption, etc. The colon cancer starts as an uncontrolled proliferation of an epithelial cell. When we take a closer look at this forming tumor, we see that it does not only consist of tumor cells, but other cells exist, such as immune cells. These immune cells can be switched off by the forming tumor, so they will not attack the cancer. During the last years, our lab and other labs, including the work presented in this dissertation, have shown the importance of the neural cells, the enteric neurons and the enteric glia in colon cancer, where they are in close proximity to the forming tumor and have impact on it. So this neuron-cancer communication has been identified in other cancer types, leading to the birth of a new field of science, the cancer neuroscience. We now know that neurons and glia communicate directly with colon cancer cells to impact cancer growth, for example. They also can communicate indirectly via other cell types, such as immune cells. We now also know that colon cancer cells hijack neural programs, granting these cancer cells neural-like properties, which enable them to keep proliferating. When I started my PhD, both these two ways weren't established in the context of colon cancer. And that takes me to the next slide, which is the goal of my thesis. So the idea of my research is to study the colon cancer crosstalk on several layers. I dedicated the first chapters to review the literature, summarizing the evidence for cancer neural crosstalk, identifying knowledge gaps and which models to use. Then I analyzed available data sets looking for neural-related signature in colon cancer as well as other types of cancer. Then I studied the impact of reduced neural cells on colon cancer in a mouse model. And lastly, I studied the opposite, the impact of colon cancer on the enteric neural cells using human tissue samples. And starting with neural signature inside cancer cells. To explain this chapter, I would need some extra background here. So for a cell to become malignant, to become a cancer cell, it needs to change its identity. To keep multiplying, to hide from the immune system surveillance, and to make friendly environment around it. These are essential aspects of cancer, or what we call the hallmarks of cancer. To do so, to have these properties, these cells need to activate specific genetic programs in their DNA. What you should know here that each cell in the body has the same information written in its DNA book. But the recipe to read out of that DNA book to make an epithelial cell is different from the recipe to read a neuronal cell. Hence, each cell needs a guide, which recipe to read, where to read, and where not to read. That means genes in the DNA needs to be switched on and off. And this can be done by processes such as DNA methylation. This process adds a specific molecule to a stretch of DNA. This will regulate the expression of genes on that stretch of DNA. And this DNA methylation changes are common in cancer and occurs early during cancer development, and hence it is used as a marker for early detection of cancer. When my colleagues were looking for markers to early detect colon cancer based on DNA methylation, they found that neural genes are the most affected genes by DNA methylation changes. And then my follow-up question was whether this neural DNA methylation signature is specific to colon cancer or inherent process in all cancers to be found in other cancers. To answer this question, I studied 16 cancer types, varying from colon to bladder to lung, etc. And we found that DNA methylation changes occur mainly in neural genes in all the tested cancer types. We also found that this occurs very early during tumorigenesis, and it enables cancer cells to acquire neural stemness so they can keep moving. In the future, we need to investigate further how this neural DNA methylation occurs in the first place. Next. We investigated the impact of enteric nervous system on the development of colon cancer using laboratory mice. To do this, we used genetically modified mice that have fewer nerve cells in the colon. We induced colon cancer in these mice and in normal wild-type mice. And then we compared the tumors formed in both situations. We firstly wanted to verify that these modified mice really had lower number of nerve cells in their intestine. So we counted the nerve cells under the microscope for each model of the mice model. And then that indeed what we found, that this modified mice had lower nerve cells in their intestine. However, the lower number of nerve cells in these modified mice had no clear impact on the forming of the tumor, the growth of the tumor, or the size of the intestinal tumors. But we did find other differences in these tumors, mainly in the tumor immune microenvironment. Our analysis showed that, in particular, the proportion of germinal center B cells, which is a type of B cells, was reduced in less innervated mice, suggesting an immune modulating interaction between these nerves and immune cells. In addition, we found that macrophages, which is another type of immune cells, were less activated in less innervated mice, so they can participate less quickly in a defense action. These findings suggest that neurons influence colon cancer microenvironment by impacting numbers and activation of certain cells of the immune system. And lastly, we wanted to characterize the neural cells during colon cancer, so the opposite, not to the impact of colon cancer, not the impact of neural cells on the colon cancer, but the impact of the colon cancer on the neural cells by characterizing these neural cells. So for this, we received resected colons of patients with colon cancer from surgery rooms. We sampled each colon in 2 regions. We took one sample from the normal part and another sample from the tumor part, and then we isolated the colonic cells. We know that these neural cells are very rare in the colon environment, so we use a technique to enrich, to isolate these neural cells apart from the other colonic cells. Then we read the active programs that are running in these cells by means of a sequencing machine that read the expression profile of these cells. And lastly, we characterized or clustered the cells according to their active programs. So we know that what cells are present in the colon and what they are doing. The isolated cells clustered indeed in groups according to their active programs, to glia, epithelia, other types of cells. And of course, because we sorted for the neural cells, the most of these cells are glia, as you see here. But unfortunately, we missed the neurons in this analysis, either because of our sampling processing or due to complications related to the length of the protocol to isolate the cells, or maybe the neurons themselves are aged and fragile and cannot survive this protocol. So we continued just characterizing the neural glia that we found, and we found 7 subtypes of enteric glia. Each one of these subtypes is doing a specific job. 3 out of these 7 families or subgroups of enteric glia had immune functions, such as introducing protruders to specialized immune cells and to prime the environment in response to the tumor. And we also found that there was a trend of increase in these enteric glia subtypes that are immune specific in cancer samples compared to the tumor, compared to the normal samples, hinting to the immune role of the enteric glia during colon cancer. So, to wrap up, a conclusion, we can conclude that enteric neurons and enteric glia impact colon carcinogenesis via immune cells, while tumor formation in the colon impacts the neural cells in their vicinity. However, future studies should be able to characterize enteric neurons in colon cancer and to develop advanced models for neural immune crosstalk in colon cancer. Thank you for your attention. I will give the word back to the prorector.
Thank you for your presentation. Your position will be opened by Professor Ruschel, Professor of Translational Biology of Vesicular Signaling at this university. Professor Ruschel was chair of the assessment committee and he is the secretary of the degree committee.
Mr. Colorectal. Well, first of all, I would like to congratulate you with this very nice achievement. Really, well, I read your thesis with lots of interest, lots of new things that I learned, because to me this is really an unfamiliar field. And obviously I would also like to extend these congratulations to your supervisory team. We all know that science is not a thing that you do just by yourself. Typically this happens in a team. I also fully agree with your proposition 8, that failure in science is not a bad thing, but it's really something that we need to progress in science. And I'm really happy to read this. And taking into consideration all the field experiments and then such a comprehensive thesis, I can only imagine what type of hard-working person you must be. So as I indicated, I've read your thesis with great pleasure and gained lots of new insight, but obviously I do have some remaining questions or some things that you could elucidate from me. You already mentioned in your presentation that in the human colorectal cancer specimens, you identified seven different subtypes of Glia cells. And to me, right, I'm not familiar with this. So why do we need 7 different types? Is there a biological function to all of them? Is that known?
Highly esteemed opponent. First of all, thank you for your generous words. Actually, for us, we found these 7 subtypes with an unsupervised clustering of these interglia. We know from literature that there are multiple subtypes of glia by morphology, by function as well. So there are like myenteric plexus glia, there's mucosal glia. So we know already 6 subtypes of glia by morphology. Only by looking at it under the microscope, we know there are 6 subtypes. And also looking at the functions that we saw in these glia, like some glia are specializing in like antigen presenting, some glia are specializing in interferon response, some are producing extracellular matrix proteins. So I think glia is doing really versatile functions in the gut. They are doing many things in the intestine, and that's why they need to have like specialization in these subtypes. That's how I see it.
Okay, that makes definitely sense. But I also read that based on the localization within the intestine, that these different subtypes are much more present or less present. But then I was a little bit surprised that in your tumor, which typically originates at a specific site within the colon, that you find all these subtypes back. Does that mean that these glia cells are recruited there, or is the phenotype of the glia cell, is that something that is dynamic? Do you have any thoughts on that?
I think this relates to the sampling. Sampling, how we sample these cells, because we took the samples as a full thickness of the gut. So we are not only taking the tumor and looking for glia and neurons inside it, but we took the tumor with the plexus under that tumor. So what we see in here, that what we name as a tumor sample, is the tumor, what is inside the tumor and what is near that tumor. And we did that on purpose because we wanted to have neurons as well. And neurons as the cell bodies are in the plexus, not in the tumor themselves. So we wanted to have this sampling strategy to get the neurons. Unfortunately, we didn't get the neurons. And that might explain why there is a little bit of like, not that too much difference we see between normals and tumors, because the tumor samples contains a little bit of the normal part, which is not normal because it's near the tumor, it's inflamed, there's many things happening, but still it might like dilute the effect that we see from the tumor.
Okay, and then if I looked at the different subtypes that you found, you find enrichment, for example, for the subtype 5, cluster 5, you call it, which is an antigen-presenting subtype. And then I was a little bit surprised, because tumors are typically selected, or they are quite pragmatic in finding mechanisms how to dampen the immune system. So why would there be a beneficial thing for the tumor when they have glia cells that are antigen presenting?
Yeah, highly esteemed opponent. I do think that there's still more experience to be done here to know what exactly this interglia is doing there. So what we see out of the expression profile of these glia, that they do express MHC2, but they lack co-stimulatory proteins. So if that's really the case, that they lack these co-stimulatory proteins, CD80, for example, then that means that they are used to dampen the immune system, as you mentioned. So without the co-stimulatory ligands, then we know that T cells will get energy instead of getting activated and proliferating to act against the tumor. So it might be a strategy indeed from the tumor to dampen the immune response against that. And that's maybe one thing, how the tumor is like making the environment friendly to it, as you mentioned.
I'm looking at the pool.
Sure.
Yep. Yeah. And then, so obviously you analyze all these glia cells and you characterize them based on these clusters, and then you say, okay, this proportion is cluster 0 versus the other clusters. But do you think it's much more important what the distribution between within that cluster is, or the absolute number of glia cells that are present within a tumor? Because I can imagine that a glia cell needs to communicate with tumor cells, and most likely it does that, and it's in near vicinity. So even if you have, let's say, 50% cluster 5 positive, cluster 5 glia cells, but only, let's say, 10 within the whole tumor, that might give a completely different phenotype than 50% where you have a really large abundance of these glia cells. So why did you choose to analyze it in this particular way?
Exactly, that's a good hint actually. Like as you saw, we did try to to correlate the proportion of each subcluster to the total number of the glia in that specific sample. Like so we made the ratio of like cluster 5 to the number of the tumor glia. But in the case, I think that you have a good point here, that we need to really count what is inside the tumor, not what is in the vicinity of that tumor. Is that answering what?
Well, it's more, shouldn't you express the number of glia cells in relation to the number of tumor cells? That's basically.
In relation to the tumor cells. Okay. That's indeed a good.
I don't know if that's technically possible.
I think like if we are going to like make the same experiment, only taking the tumor, that would be possible definitely. Then we know the exact proportion. But now, because we have together these like tumor cells and what is in the vicinity of these tumor cells in one sample, I think it will be hard to do it in our setting. But in another setting with only tumor cells inside, I think it should be beneficial to have a look at this, indeed.
I think I'll hand the word back to the board.
Thank you. The opposition will be continued by Professor De Jonge, Professor of Experimental Gastroenterology at the Amsterdam University Medical Center. Professor De Jonge was member of the assessment committee, and we highly appreciate your participation to this academic ceremony.
Thank you, dear candidate. I'd like to extend my congratulations to the thesis that you have produced. It is a lovely piece of work with lots of disciplines. Innervation of the GI tract is a complex matter, and especially if it comes to colorectal cancer and how this is developed. But I also have a number of questions actually that relate to, let's start with chapter 4 and figure 1, which is on page 74. It illustrates a nice picture with the extrinsic innervation, I believe, extending towards epithelial cells. For instance, I see there acetylcholine coming from an extrinsic neuron. Do you have information of evidence that cholinergic neurons that are extrinsic actually innervate cells that are non-neuronal in the guts, meaning epithelial cells, tumor cells, any type of cells. Do you think that's existing, or is this a cartoon to show an idea?
Highly esteemed opponent. I think when I had this figure drawn in that review, I think I revised all the the literature that was there, and I'm not really 100% sure if that was the case, but I think I did it on purpose to have extrinsic peripheral acetylcholine nerves, but I'm not really 100% sure anymore. I know, for example, that there are some sensory neurons that are really from the central nervous system coming right away, but exactly with acetylcholine, I'm really not having this information in my head now.
That's a nice answer, and I think that is correct. We're not entirely sure, but as far as I know, there's no evidence for extrinsic innervation through cholinergic vagal neurons that would release acetylcholine. But there is a lot of receptors on epithelial cells and adrenergic receptors on epithelial stem cells as well. And that kind of refers to my second question. In one of the discussion points in this chapter, you also mentioned adrenergic receptor activation breaking down p53 and limiting apoptosis. That's on page 84, I believe. And my question is a bit, so it's figure 3. And my question is a bit, how is this actually possible if these stem cells are innervated by adrenergic neurons? And adrenaline would be that toxic to cells that there will be a, let's say, a degradation of p53 and a pro-cancerous effect of that innervation. How is that even possible then, if those cells are actually innervated?
Yeah, highly esteemed opponent. Maybe just to let me rephrase the question. So you are asking that degradation of p53 is toxic to the cells?
Well, I'm referring to page 83 on top of that page, and you can be short on the answer. Adrenaline is shown to stimulate degradation of p53 and the production of ROS, thereby leading to DNA damage. And I was just looking at your figure 1 and then trying to understand what you write here. But how is that even possible if cells are that are in the stem cell niche are actually innervated? And you described that is a pro-tumoral oncogenic event, that innervation. So I'm just wondering how that works.
So highly esteemed opponent. So I would assume that there is a kind of balance when from norepinephrine or doing specific functions, also from the parasympathetic input, also from the input from the ANS, there should be a balance that is regulating the proliferation of these specific cells. When we have extra signaling out of a specific input, for example, beta-2 adrenergic activation, and that's what is done in that experiment, then they saw that there is indeed a p53 degradation and more reactive oxygen species production that might also promote tumorigenesis in a specific way. So I think it's the imbalance that is in charge here more than the balance that is in the gut in the first place.
Yeah, well, it's a complex matter. I agree with you. So maybe we should move on to chapter 7 then, because I'm looking at the time. I have a more or less more specific set of questions. I'm really excited about this chapter. I think you've done a lot to show that these specific subsets of glia are there in the human context, and you just also commented on questions that were raised on the previous opponent. My question to you now is one that relates to the way these cells were isolated, CD90 and another marker, NMAM1. How sure are you that these are glial cells and not other cells like fibroblasts, for instance, or stroma, other types of stroma? How specific is that marker set for you?
Yes. So for this for this protocol, the enrichment of neural cells, the protocol is developed by the lab in Rotterdam, where I also work in. My colleagues worked hard to develop this, and they had validation. Although they developed this protocol on pediatric tissue, they also published the protocol. As a protocol, how they are sure that these are neural cells. They also had like single cell data showing that what they are getting from this strategy of enrichment is indeed neural cells, and that was also the case in my data. So when I did the single cell experiment afterwards, it came out that these are mainly glia cells. So we see the enrichment also afterwards, that in the single cell experiment.
And these are not fibroblasts.
So according to the active transcriptomes that is in these cells, they are glia. And to compare it with, I think there is a small proportion of fibroblasts. You can maybe see it in figure 3, that we have a little bit of fibroblasts in there.
I agree with going there, because that's exactly what I wanted to move on. In figure 3, actually there is quite an interesting group of fibroblasts that are CD90 NCAM negative. Indeed, those are EC gels of fibroblasts. If you look at the stroma versus normal, that's where the difference is at. Not do I understand that correctly? In 3C, figure 3C. I'm looking at tumor versus normal, and then the expression differences in, for instance, the group of cells, it's a bit small, that is EC or fibroblasts, is interesting, especially in the lower fibroblast tumor versus normal. That's where the biggest difference is at, or do I not understand that correctly?
I agree with you that we did see indeed big differences between the normal and tumor samples in some of these, what we called contamination of the sample, because they are not neural, including this fibroblast. And when we verified that, okay, what are these differences in details, but it's not in the written in the chapter, but we when we verified, we saw that these differences goes in agreement with what is published already about fibroblasts, tumor-associated fibroblasts, and other types. Like we see also such big differences in the epithelial, because we do have tumor cells in the tumor and we don't have these tumor cells. So that was also a striking difference. I think that's what you hint to.
Yeah. Related maybe a bit to that is the population of GFAP positive cells that you also describe in your discussion, that you could not pick up. I think that's an interesting finding. My question to you is, do you have explanation for that? I also saw that there were 8 glial cell subsets, of which one was expressing a high level of mitochondrial DNA and probably dying. Could that be this GFAP population?
Yeah, that's an interesting suggestion. I think I did. So when we removed that one, I did the analysis several times to make sure that I'm getting an optimal overview of the of these cells. I'm not really sure if I did the GFAP like feature plot or violin plot before removing that type of cells, but I think it's worth rechecking again that these are not the GFAPs. But the thing is that those ones, the cluster that I wanted to omit, was high in mitochondria, high in like heat shock proteins, high in like really signs, like also in ribosomal percentage. So really signs of dying cells rather. But it might be that these GFAP positives are more fragile and they are more likely to die. That's indeed a good suggestion that I really want to look at it again.
Yeah, and maybe spatial is a technique to use for this type of analysis, not?
Definitely. I think it's the next step is to go spatial, because we of course missed the neurons. That was one thing that we really saw as a pity, and we also see that there are, like, we now know that there is like several subtypes of glia, but we are not sure if they are really interacting with specific tumor cells or with specific immune cells. So spatial proteomics and transcriptomics would be really much valuable in here.
Yeah. I would like to thank you for your answers. If I look at the time, I have to stop here, but I enjoyed your reading your thesis a lot. I'll hand over to Director.
Thank you. The opposition will be continued by Professor Beeson, Professor Experimental Vascular Pathology at this university. Professor Beeson was also a member of the assessment committee.
Thank you. Dear candidate, first of all, my congratulations on your thesis. It's a very nice piece of work, and also commend the supervision committee. It's a very intriguing topic, and I'm completely naive here in this field, but I got the impact of neurons on carcinogenesis, and it's I think there's much more to follow there. So. As I told you, I'm not an expert on ENS, and it even confuses me to be aware that I have a second brain while I'm only hardly able to handle my first one. But it's good that the gastrointestinal tract is also taken good care of. So I would like to follow up to the reference of my colleague, Professor Rushop, on Proposition 8. And I'm curious here, what is the most defining failure in your studies that led to major success?
So yeah, highly esteemed opponent. Thank you for your nice words. So there are of course many failures, setbacks during this whole process, specifically because of Corona, where it hit, and
that's not a failure.
It had a huge impact on my research flow, because my research started with pluripotent stem cells, with making organoids, modeling cancer, but with all the lockdowns, and had to stop like these long-term cultures. So I needed to really switch away and pursue other types of projects. But that was one thing that I really needed to have some
the outcome, and
yes.
So you have to be commended on your flexibility there. So my scientific question related to question 1, so chapter 4, and of course you're probably aware that my major interest or my pet cell is actually the macrophage, and I looked into figure 3, and I was surprised to see there's a macrophage in. There's a direct interaction between the neuron and the macrophage, guided by the neurotransmitter noradrenaline, that apparently skews macrophages towards a TAM-like phenotype. This is always the case, by the way, is that adrenaline is sort of polarizing towards TAM-like M2 cells.
Okay, highly esteemed opponent. Actually, there's, let's say, some in vitro data showing that noradrenaline is indeed promoting carcinogenesis, either directly on the cancer cells or also on the indirectly via these, as you see in figure 3, not only macrophages but other types of immune cells. Actually, if I get your question correct, do you mean
if you expose macrophages to epinephrine?
Yes.
Is it always skewing towards an anti-inflammatory phenotype, or is that depending on conditions and on the phenotype?
So the studies that I've read was specific to the macrophages that are resident macrophages. So isolated macrophages or cell lines, macrophages cell lines that is treated with norepinephrine. But I don't know what would be the response for example, for lung or for spleen macrophages if they are treated with norepinephrine. I would assume that it is similar, but that needs, of course, experimental proof.
And how does it work? Is it signal transduction mediated by the beta adrenergic receptor that is inducing this, or is
So
interference with?
So it's also as in figure 3 that these macrophages has beta adrenaline had beta receptors that norepinephrine can work on, not only noradrenaline but also adrenaline. So I think it is direct interaction between the neurons and the macrophages, but I think there is also a chance for indirect communication between these cells, like norepinephrine. I think like via glia, like if you activate glia will secrete, for example, interleukin 6. This interleukin 6 will activate macrophages to become also M2 phenotyped and to also make them tumor-associated macrophages. So I think there's 2 or many ways for these macrophages to become activated, not only direct communication.
Yeah. And if I compare that to the other route of neural regulation mediated by acetylcholine, I don't see macrophage anywhere. So does that mean, is that an artistic freedom that you took, or is that is that a deliberate choice to leave it out here in figure 2?
So again, the idea of these concepts is just to take, so now you see like acetylcholine, for example, and noradrenaline in figure 2 and 3, but that doesn't mean, like, for example, serotonin is not doing anything. It was a choice that I pick some of these neurotransmitter that is doing. If I, this is 4 years already, but if I'm recalling well, if I found any piece of evidence saying that macrophages are, in the context of gastrointestinal cancers, would get activated by acetylcholine or deactivated, I would have put it in there. But I think that's the case.
Okay, good. Now then I move to, if time permits, I think there's a bit of a few minutes left, on to chapter 6, where you studied the interaction between neurons and colon cancer cells in hypo versus normal innervated mice. You probably were a bit disappointed to see no effects, direct effects on tumor size or tumor characteristics, but you can also appreciate this as an advantage because your subsequent RNA-seq were not biased by any compositional changes, so that any disadvantage often leads to some advantage. So looking into your outcome here, you see that you have increased DNA repair, at least GO terms enriched that point to increased DNA repair, cell cycle, and the suppression of immune profile and EMT in hyperinnervated mice. How would you, what would be your strategy to verify this experimentally?
Okay. Highly esteemed.
Because the limitations of aura, the enrichment analysis. Not always it gives you clues, but no direct conclusive evidence. Huh?
I think we have here like some things to hint. Like, and although there is like significance for these specific like gene ontology terms. But if you looking to the figure 3a, we really had very little of genes that are hyper expressed. So that means we can at first step, looking what can be verified in here. At first step, we need to verify or to validate that these genes are really upregulated. That's one. So this is expression, and we need to validate. Actually, we tried specifically for MBY to validate this with like culturing specifically on the outcomes that come later, not specifically to the DNA repair and those like MYC targets and others. We tried to make like conclusions specifically to GC B cells, the German center B cells, if this had some impact on it. Actually, we find it more starking differences with norepinephrine treating these immune cells compared to MBY. Actually, it should be one of the future experiments, like to really look at these specific pathways inside cancer cells, like what is the impact of hypo-innervated colon on DNA repair or these things. But this is something really need to be done in the future.
So, future research.
Exactly.
Thank you. I'm fully content with your answers, and I give the word back to the pro-actor.
The opposition will be continued by Dr. Wolf, Associate Professor of Perinatal Medicine at Maastricht University.
Thank you, Mr. Pro-Rector.
Wolf was also a member of the assessment.
Thank you, Mr. Pro-Rector. Dear candidate, obviously also I would like to congratulate you with this excellent thesis. I read it with a lot of pleasure. I also would like to congratulate your excellent team of promoters. I would like to start with a question about one of your propositions, and I would like to ask you if you could please read proposition number 7.
Artificial intelligence will revolutionize omics analysis for uncovering patterns shared across different biological layers, such as genetics, epigenetics, and proteomics that are beyond human perception.
Thank you. I'm really interested about the rationale behind this. So how do you foresee that AI or bioinformatics, which seems to be a topic where you are very good in it, will be the game changer for the CRC domain and beyond?
Highly esteemed opponent. I think like now we are living in the era of artificial intelligence. Like every single sector is benefiting out of it, and we as biomedical scientists should really be using this technology. Now we have, like, for example, several layers of information. Like we have the transcriptomic layer, we have the proteomic layer, we have also a lot of imaging that we need to analyze. Actually, if we take like conventional ways of data analysis, you need to come with a hypothesis. You really test that hypothesis as you see it as an analyzer. But if you give the task to AI can comprehend, I hope in the near future, can comprehend what are the hidden clues in between these all layers. Like now we can, for example, need some data in transcriptomics that we need to verify in proteomics, and then we can look at it in other layers. But like we can ask AI to, okay, find together all these data in one shot. What do you think is happening? He can combine in one shot, like as if, yeah, I don't know if I'm really enthusiastic about the AI here.
I can see. And I assume that you created already a basis for this with the work, among others, described in your thesis, right? Okay, having said that, the work you've done focuses on the role of TNS and colorectal carcinogenesis. Development of TNS starts in principle early in life, actually being in utero. And it's a tightly regulated process that can be disrupted if there are suboptimal conditions in early life. If this occurs, this might lead to Hirschsprung diseases, necrotizing enterocolitis, and also potentially food intolerance. My question is, can we learn lessons from the CRC domain for the developmental biology field, and the other way around?
Nice. Highly esteemed opponent. I think this is an interesting topic that we need really to focus on. So the thing is that we have really some data showing that early life events can impact colon cancer. Like eating very bad diet during childhood will make more susceptibility for colon cancer. We don't know why, but at least we know that it is epidemiology the case. Also, there is a Dutch, I think, like the big famine that happened 60 years ago. They also saw that during that period, people who lived in that famine situation had altered susceptibility to colon cancer. I think that that should relate to how the cell get predisposed to cancer. Like, we know that cells have memory, like not only the DNA methylation memory, but also immune cells have memory. They stay with a person to his life. So I think there's something interesting. Let's say talk about DNA methylation, for example. Like there are experiments that, like from organoids, you keep those organoids proliferating for 1-2 years, and then after 2 years, spontaneously they get cancer phenotype because of DNA methylation changes happening because of BRAF mutation. So I think we need to verify what is exactly happening during early life and how that will impact either DNA methylation or the immune memory cells, and what can be modulated in the meanwhile.
Yeah, I'm really pleased with that answer and intrigued by it. What about the microbiome, for instance? Do you know anything about antibiotics treatment around birth and impact on immune cells, or the interplay between the ENS and the immune cells?
I think microbiome is an interesting topic to look at with this early events of life, because at least like we know from other studies from other fields that, like, for example, antibiotics that is taken by the mother is impacting many diseases in the progeny, such as asthma that was I involved in years ago. And I think it is like one level of information that really is interesting to look at, specifically in colon cancer. I don't think there is now data showing a correlation between early event lives, microbiota, and colon cancer, but I think it's worth investigation in the future.
Is there room for a small question?
Short question and short answer, please.
Yeah, so I don't know whether it's a short, smart question. So,
but that's okay.
How would an ideal experiment look like if you are able to do a follow-up experiment, and if you focus particularly on an intervention, because we didn't talk about interventions? On what intervention would you particularly focus on?
Based on the early life events of
exactly, yeah.
I think we need to really look at the DNA methylation, because it's very early. We can, if we can intervene in this step, or even before that, if we see that like intervening with DNA methylation is something that is, I think, interesting. I hope that's a good answer for a short time. But
I think that's a perfect answer for the room of space we have. Yeah, thank you very much for your answers. I give the word back to you.
The opposition will be continued by Dr. Cirillo, enteric neuroscientist at INSERM at the University of Toulouse Paul Sabatier in France. Dr. Cirillo was also a member of the assessment committee, and we also highly appreciate your participation to this academic ceremony. Dr. Cirillo.
Thanks, it's my pleasure, actually. First of all, I want to thank Mr. Candidate for sending me the book of your thesis. I received it yesterday evening. It's a pleasure, and I really love the I really love the cover. So I have just a few points I would like to discuss with you, because other questions have been already asked by my colleagues. If we go to chapter 4, indeed, where you describe it's the interesting review you drafted about the denervation that has been done to see the impact on the progression of colon cancer. So there are, let's say there is still contrasting results on this. In your opinion, and based on your, your knowledge, there is a way, instead of acting on the innervation, on acting on the glial side, of, yeah, in this context?
Highly esteemed opponent. Do I get your question correct if you mean that you are asking about something else other than denervating the intestine?
Yeah, I mean, shutting down the glial component.
Yes, I think denervation is one way that is like done in the surgery rooms for specific patients, specifically for ulcer, and that's why it is it was worth studying. But I think it's more important to have more specific intervention with these patients. Something like interfering with a specific component, such as, for example, downregulating, for example, neurotransmitters that we know that they are priming immune cells to not to interact or against this cancer. Also in Glia, because it's a hub of activating or a hub of immune responses in the gut, it's also likely that the patient will benefit of manipulating these pathways. Some interleukins that we know that they are priming some immune cells for being like pro-tumorigenic. Also, the glia itself, like secreting these Wnt ligands, is also something that is interesting to look at. And really, if we can develop an intervention specifically to these neural cells, that's actually the aim of this understanding of the field.
Okay, thanks. And continuing indeed on your question and going more in detail, based on the signature, the neuronal signature that you found in your chapter 7, the work in chapter 7, do you think that this can be like a future therapeutic approach in CRC? Instead of acting, you know, on the epithelial component, but maybe on the neuronal component?
I do think that but sorry, you mentioned the signature in chapter 7 or chapter 5?
Chapter 7.
So the antigen presentation and the glia impact. I do think that cancer is a heterogenic disease that needs to be approached from several sides. Like, although glia seem to be deeply involved in colon carcinogenesis, but I think many other types of cells are important here. So I think the best strategy to treat colon cancer is to address several sides of the disease, not only glia, but also the epithelial cells themselves, also the neural input, like with the neural derived drugs. So and also that all belongs to specialized, to personalized medicine. Like each patient needs a specific, so there should be like stratification also based on the density of nerves, maybe in these patients, the density of GFAP positive glia in these patients, to know if this is, if the like counteracting glia pathways would be beneficial for such patients.
Okay, thanks. And an information that I could not find, always related to chapter 7, in your cohort of cancer patients, did you have different stages of cancer? And if yes, could you see differences in the signatures that you found?
Yes, actually, the thing is that we didn't have an exclusion criteria here about the stages, but the material that I received was always late stages. So so always big tumors that are really passing through all the layers of the intestine. So on the other hand, we also didn't have exclusion criteria on the segment of colon that we are testing in here. So like ascending, descending, also sigmoid is involved in here. But mainly this data, because the samples that we got mainly came from sigmoid colon, so mainly this data comes from sigmoid colon piece. But the other segments also included.
Okay, thanks for the explanation. From my side, I don't have additional question. I just would like to finish saying that, I mean, after what I heard for the COVID situation, I think that you have learned not only to be a scientist, but also to be a flexible and adaptable scientist. For sure, thanks also to your supervisors, but this is something that I'm sure you have learned during your PhD program. Thanks a lot.
Thank you.
Thank you. The opposition will be continued by Professor Rushop.
It's me again. Hi. So I'm happy that I'm able to ask a few more questions. So one of the things that, from going back to my last question, was basically on the distribution of the different clusters within your glia cells, let's say. And your conclusion is basically that cluster 5 increases. But if I look very carefully at your graph, then you could also make the conclusion that cluster 0 is actually decreasing. And I'm definitely not an expert in your field. I'm very ignorant, to be honest. And coming back to the antigen presenting characteristics of cluster 5, then so in my opinion, that could also be then, let's say, an anti-tumor cluster. But what if your cluster 0 is actually the tumor repressing cluster, which is decreasing in your tumors? Is that a possibility? And do you know anything about the characteristics and the phenotype of cluster 0?
Yes, thank you. So, highly esteemed opponent. I do agree that there's downregulate or less of cluster 0 and a little bit also from cluster 1. We definitely.
Hora asked.
You can shortly finish your answer.
Yes. So, this is a proportion. So the cells that is getting less from cluster 0 is going to cluster 5. If it's only cluster 0 that is moving away, then it should, I think, being spread in all the other clusters. That's one side. And another side, that cluster 0 is basically the baseline glia. That's the main characteristic of this cluster. So it is, I think it is more likely that cluster 5 is the more interesting in here.
Thank you.
Thank you. Musa Idris, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense, and I request that you and your company await the results of our deliberations and our return to this room.
Please all rise. Musa, please come with me. Paranems, please follow. Jullie mogen binnen.
Musa Idris, the degree committee here present has assessed the quality of your thesis and your defense. In view of its positive verdict and taken into account your previous qualifications, the degree committee has given you the degree of doctor. Dr. Milot is authorized to confirm upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor to now take the floor.
Please all rise.
Do you promise to work in accordance with the principles of scientific integrity in all times, to be careful and honest, transparent, independent and responsible?
Yes, I promise.
By the authority vested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Musa Ahmed Idris, the degree of doctor and grant you all the rights attached to custom and law. As evidence of this, I now present you with the degree certificate signed by the rector, the secretary and the other members of the committee and affixed with the official seal of the university.
It's a big one. Thank you. This is for hours here. Als wie كيف لابسين هي.
Please be seated.
روح عادي خلاص روح عادي. روح عادي بكير ماشي.
Oh, you want to take it?
I can.
Selena, one hand in the middle, underneath.
One hand and
Yes, very good.
Okay. Esteemed Dr. Idris, dear Musa, from today you proudly may call yourself doctor, and I'm deeply honored to be the first to congratulate you also on behalf of Maria and Werend. Of course, I would also like to include your wife, son, other children, parents, family and friends in these congratulations. Many of them are likely celebrating online or currently elsewhere, sharing in this milestone moment, both for your PhD but also for your homeland situation. Your journey with our group began in August 2018 already, when our grant officers introduced us via email with the opportunity to apply for an NWO Hestia grant together. A week later we met, and the rest, as they say, is history. I think I can say we clicked from day one. Our conversation covered everything from global human rights and politics, but luckily we also find some time to discuss science and to write the NWO research proposal together. I still remember our train ride from Utrecht after the NWO interview, where I had to encourage you to take this opportunity, a grant specially designed for refugees, as you were hesitant to be seen differently from non-refugee scientists. You performed very well, and soon after, we received the fantastic news. The fellowship was awarded, but unfortunately only provided funding for 2 years. As you were offered a PhD position elsewhere, I knew I had to come up with a plan. So after knocking on many doors, also in at Maastricht University, and getting no for an answer, I reached out to Professor Robert Hofstra at Erasmus MC in Rotterdam, whose love for creative, out-of-the-box solutions, especially for young, talented scientists, led him to offer you support for a full PhD track. With additional backing from our research institute GROW, we were able to launch your PhD journey, giving you affiliation at Maastricht University and also Erasmus MC. Living in Eindhoven, right between Maastricht and Rotterdam, you traveled a lot during these days, which is already a testament, I think, for your dedication. Tragically, Robert passed away in 2021, but I'm grateful that you, Werend, Maria and I had the chance to work with him and to learn from his leadership and mentorship, which I hope we can all put forward in the future. Maria, your co-promoter, continues Robert's work, ensuring that your PhD track could progress smoothly at both universities. I'm sure I can say in the name of all team members that you have been a truly valued member at both Maastricht University and Rotterdam. You brought a lot of knowledge, expertise in bioinformatics, but also delicious Syrian specialities to the table. And I believe this is also thanks to your wife. And I'm not sure which skills were appreciated most in the group. In a group with predominantly female colleagues, your calmness also stood out. I never once saw you lose your temper, not even in moments of high stress, for example, during intense meetings with me and Maria. I think the work you're collecting during your PhD has been groundbreaking in the emerging field of cancer neuroscience, and the inclusion of Weren Boesmans as a second co-promoter added essential experience in the enteric neuroscience. I'm also convinced that you were very happy to having a male addition in your supervision team. I'm proud that the three of us, coming from different backgrounds, were able to combine our strengths and to bring our PhD track to a successful end. Next to our supervision, you collaborated also with many more international researchers, so as Dr. Max Maé from the University of Nantes and Professor Tim de Meijer and Dr. Louise Cosemont from the University of Ghent, which further enriched your skills and broadened your network. Musa, I have been focusing on your strengths so far, but I believe I must also mention your chaotic nature, which I think all of our group members have become familiar with, either with working with you in the lab or by organizing meetings together. As a fellow chaotic soul, I must say that it was rather challenging towards the end of your PhD, when you insisted on defending in 2024, despite the last-minute rush. Until just 10 minutes ago, I was anxious hoping that all forms and regulations were in place to ensure that you would receive this PhD certificate. And I must also deeply express my gratitude to the committee members who are here to make this possible. Normally, as many of you know, I'm a quite demanding supervisor. But Musa, you somehow got away with things that others wouldn't. I will miss our conversations about science, but also about human rights and global politics, and I sincerely hope we stay in touch. Maybe one day, one of our team members will also find themselves in need with your smuggler talent. Honestly, I hope everyone in life gets a chance to meet a Musa, to learn the immense value of diversity and to feel the joy of helping each other, and to see how solidarity can overcome the biggest human challenges. Musa, you have come so far, and we are incredibly proud of you. From today on, you can call yourself Dr. Idris. This is because of your scientific expertise, your collaborative spirit, your persistence, and your hard work, and not just because you received the Hestia grant. Last week you told me that you didn't really have something to celebrate, but I hope you have today. This PhD track is done. On behalf of everyone who has been part of this journey, congratulations on this well-deserved achievement. We wish you, but also your family, friends, and considering what has been happening over the past days in your homeland, a future full of peace, happiness, and new opportunities. Thank you, Musa. Thanks for joining our team, and it was an honor working with you.
Dear Dr. Idris, yes. You cannot reply anymore. Also on behalf of the Maastricht University and the Board of Deans, it's a pleasure and an honor for me to congratulate you with this new distinction as doctor. I believe it must be a special and lifetime event to defend an academic thesis with all the ongoing developments in your country and in your hometown. So we wish the whole university, I believe, wish you all the best in the future, and of course also with your academic career.
Thank you so much.
And I will now close this academic ceremony. I will ask the audience, but of course not the family, to leave the room. And we will make a picture in front of the screen with our online colleagues of the degree committee, and afterwards we will take a picture on the stairs in the central hall. Okay. Here I close this academic ceremony.
And you are invited for a Syrian reception outside.
Yeah, the reception is in the museum here, huh?
And the